Okay, so there we go. My camera feels weird. Okay, so me and about like 80 other people just finished their first ever grant proposal. It's not an actual grant proposal, but it's like the introduction and specific aims page. And so basically we just had to come up with a project. It had to be related to uh, like cell and development biology. And so I chose the role of linker of nucleoskeleton and cytoskeleton, also known as LINC complex, and nuclear polarization in migrating fibroblasts. So this is just chapters that we're going to go through today. So the longest one is number 1 because it's background. I don't expect anyone to actually know what a fibroblast is. And so we're going to talk about that, and then my actual hypothesis and how I set up my experiment. So cells move. Shocking. So cell migration is a necessity for tissue repair and homeostasis. Fibroblast cells migrate to a specific part of the body. And so in order to do that, they need to be polarized. So polarization is defined as a cell having an apical and basal ends. So like a top and bottom end of a cell. So um, impaired cell migration, like say the cell is not polarized, it can't move, and so things like that, impaired cell migration contributes to complications such as poor wound healing, scarring, inflammation. I'm going to put all the references in the description just so people can look them up. But okay, so here we have, I'm not sure if you can see my pointer or not. Let's, pointer, there we go. Okay, so here we have a static image of a fibroblast cell. Basically, they're cells that hold tissue together. Your muscles are made out of them. And they're super important, right? Like your whole body, basically. Um, and so when they move, it's usually in response to a wound. So burns that penetrate like multiple layers of the skin, second degree and third degree burns, there's like a huge freaking gap where there's no cells. And so the body, like damaged cells or immune cells, release signals to the surrounding area, like tissue area, and they're like, come, help, help. Uh, and they make bridges and fix up your wounds. So wound healing takes a long time, but we'll get into that in the future. Okay, so moving on. This is a diagram showing the polarization that a fibroblast cell has. So sensing direction, there's damaged cell stuff floating about. They're going to sense this, and then they're going to start actin polymerization at the leading edge of the cell. So like this would be the apical end, and then down, so apical or cell front, and then basal is cell rear. And for this polarization or organization, asymmetric organization in the cell, well, I guess it's yeah. This is super important. So we have the nucleus at the rear, And then we have protrusions. We have actin polymerization happening here. So the cell is pushing towards that, it's grabbing onto the substrates around it and pulling itself along. And then in the cell rear, we have contractions happening. So they're detaching from the substrate and like pulling in. So you get that movement. And so this is a video that I just freaking love. It looks like a little monkey like climbing through like a net. So the green here is extracellular matrix. I'm not exactly sure what protein or complex it's marking, but the purple thing is the cell. And then I'm not sure if you can see, but there's also a blue circle in the cell. Let's see. I don't know if I can catch up with it. There. That's blue. That's the nucleus. So, and then over here we have cell sensing, and then the nucleus also reorientates itself a little bit. So cell movement, I just talked about this. Wound healing could be greatly increased. Like the time, like if we could figure out what draws these cells or how to make them move faster, maybe we could have treatments for specific wound healing, especially when it's like dire cases like third-degree burns. So super important, but also what other cells move? A bunch of other ones, but metastasis is a huge problem, right, in cancer. And so if we could figure out how to slow them down or stop them moving, that would also be great. So, yeah, so you should care, basically, what I'm saying. And also, just, ooh, I guess I can't go back. Also, just to know more about cells is cool, so that's another reason you should care. Okay, so in cell movement and polarization, we have this reorientation of the nucleus. So it's a bit hard to see in this video, but Manonov 2013, they did this study on cell fibroblast migration, and they were able to mark one end of the cell, or one end of the nucleus, to know, I'm pretty sure it's the microtubular organization center that they marked, but I'm not 100% sure. But just so you could tell, like the nucleus is one of the, I think it is the stiffest organelle in a cell. And so it's like really tight. So a lot of these studies that look at cell movement have cells move through pores. So, and it's really cool. In other cell types besides fibroblast cells, the cells will actually choose the path of least resistance. So if there's a hole that's bigger, it will go through that. I'm not sure if you guys have seen like cat videos where they have to jump through a hole, whatever. And so the cell orientates itself so that the nucleus, like the x-axis in an oval, if it's like a vertical oval, the width of that is like the smallest that the nucleus can get. And since you can't squish it anymore, it's super stiff, the cell, what pores the cell can get through are dependent on the nucleus and its size. So here we have this So basically, what this paper showed, or whatever I got from it, was that the nucleus is kind of like a steering wheel in a car. And so it will get signals from like the leading edge or wherever, you know, the damaged cell molecules are coming from. Be like, okay, I got to get there. Somehow it gets the signal that, oh, we should be over there. And so the nucleus will turn and then the rest of the cell will follow. Um, and so it's just really cool, right? Like you don't get perfect reorientations, but it's pretty great. It's like really interesting. And so I have this hypothesis. I wish I had a diagram. I don't have a diagram, but I hypothesize that the linker of nucleoskeleton and cytoskeleton complex, or LINC, is responsible for the rotation of the nucleus, so this reorientation. Reorientation implies that there is something to like orientate. So my second hypothesis is to mark that, be able to mark what the apical and basal ends of a nucleus is. So it's not really known if the nucleus itself is polar, like the rest of the cell around it. We need to study that. So this is what I'm proposing. My experimental setup will be, so I'm going to use, this is all fake because we're not actually going to do the experiment, but say I would use the rat2 fibroblast cells. They're really well suited for this study due to their robust responsiveness to experimental conditions. They're resilient in culture, and they're really easy to induce mobility. So, and then FRAP is an interesting method. So, I can't go back. So you remember how we have to somehow mark the nucleus to know what side it is, right? So we need to either mark the microtubular organization center, which is like in front of the nucleus. We could also do this FRAP method where we induce like a spot that gets bleached. So inside the nucleus is this matrix or network of like a skeleton of lumen and chromatin and histones. Like I said, it's super dense. And so if I were to do this, I would bleach one side of the lumen inside the nucleus. And hopefully, like, since it's so stiff, I don't think there is going to be any weird like membrane diffusion or stuff like that. Maybe there is, but photo bleaching should just mark it. So there's probably other videos on FRAP, so I won't go into it too much. I also don't know that much about it, so I would rather stop talking about it. So to visualize this, so the microscope that I want to use to do live cell imaging is like a confocal microscope. You could also do it with just like a light microscope, but I'm not sure how you would do that. So you label proteins in the cell so you can see how things move. So actin here is getting labeled because it defines what the leading edge of the cell is doing and what the contractions at the rear of the cell, what's going on there, right? And then sun is a inner membrane bound protein in the nucleus. So the nuclear envelope has sun proteins. They have a bunch of other proteins. This is why I wish I had a diagram. But the LINC complex that I mentioned earlier relies on the sun proteins. So without the sun proteins have to interact with another protein that has a cache domain. Forget what it stands for, but once they come together and they bind, the sun protein is connected to the cytoskeleton outside of the nucleus. So it's like a transmembrane protein. And on the inside, sun is like grabbing onto the lumen, like the fibers in the nucleus. And so it's literally linking both the cytoskeleton and the nucleoskeleton together. So I'm marking that because it's important. And then dyene was a thing I threw in at the last minute. And I wanted to, literature has shown that like dyene is important for the reorientation of the nucleus. And so, but the thing is, dyene is across the whole cell. So I want to label the new dyene around the nucleus. So as the cell rotates, I want to see that dyene. And so to do that, I'm going to label this, literally can't see it. Oh, fuck, what's it called? BICD2? Is that right? Yeah, BICD2. So this protein is a recruiter of dyene and a bunch of other stuff. So that's related to the cytoskeleton. And so by labeling that, it will give us sort of an idea of the new dyene or dyene polymerization induced by that recruiter. So it's all fake. It's just a project. So how will you actually get like quantification? So there's a bunch of different live cell image trackers out there. There's the ImageJ, which is extremely important in like biomedical science, or it's just biology in general. Like, you guys should check that out if you haven't already. You have to, if you're gonna do imaging and publish it, it has to go through ImageJ for that to work. So it's like you can't just like upload a PNG or whatever. Yeah, so that's a separate lecture. But there's this cell profiler. This is just what I literally just Googled tracking methods, and this one seemed to fit what I wanted to do. So cell profiler, it's open source, great. And then it does the cell tracking, so live cell movement tracking. And then it also has features where you can mark specific sections of the cell and track that. And so this is actually like uh, a subset of like machine learning. A lot, I think people use this for training like large learning models on like prediction of where the cell is going to go. But anyways, so the tracking software will be used to test for correlations between the areas on the cell membrane and areas identified on the nuclear membrane. So like the sun proteins. So my first aim is to assess the role of the LINC complex in nuclear repositioning. This aim is supposed to determine if that LINC complex, if disrupting that LINC complex affects the nuclear rotation. So um, if the LINC, if nuclear rotation is LINC dependent, um, we would see we wouldn't see rotation if we disrupt the LINC complex. So in the case where it's LINC independent, that means that even though we fuck with the the LINC, it's still rotating. And so if that was the case, it would exclude the LINC complex from being the direct, thing that's rotating the nucleus. But other uh, previous research has shown that disrupting the LINC complex disturbs cell mobility. So if nuclear rotation is LINC independent, it could LINC could still influence cell mobility in other ways, motility in other ways. So this image here, uh, this lamina, I think that's like the network. It's made out of lamin, like A and B. Sun proteins bind to that, and then they bind to other proteins that are on like the outside of the nuclear envelope. Uh, you can also, this is continuous with the ER membrane. And so just to help picture that, so there's this cache domain that the sun protein has to interact to connect to the cell, the cytoskeleton. So here we're gonna disrupt the LINC complex and okay, so like here, this cache domain, um, this whole protein in mammals, they're called the Nesperin proteins. So they interact with the sun proteins and connect the cytoskeleton to the nucleoskeleton. So to disrupt this complex, I'm going to construct a construct, a protein construct with the cache domain as the only thing that it does. And so I'm not exactly sure what protein I'm going to like create a cache. I'm not sure, but I know I can do it, and so it's out there. So if I make this cache domain protein that is basically a cap for all the sun proteins. So it will, if there's high expression of it, it will outcompete all the other ones, the Nesperin proteins. So sun proteins are no longer able to bind with the Nesperin proteins and have this LINC complex because they're already bound to this nothing protein that we made. Um, and then as a control, you want to make the same kind of protein, the same construct, but with a non-active cache domain. So just to make sure that everything's, thought of. Um, like I mentioned earlier, the LINC disruption of the LINC complex has been linked to lower cell motility. And so to verify that we did mess with the LINC complex, uh, we can see we should see that there's lower cell mobility in our samples. That can be measured by what I mentioned earlier, like the pore migration. So like my cells migrating through a filter, uh, and you just like measure how many cells got to the other side at a certain time point. Uh, yeah, so I think we covered all of that. My aim two is to determine whether the nucleus polarizes during cell migration. So, like I mentioned earlier, we know that cells are polarized, and this is really important for function. You can think of like neuron cells or like gut cells, like in your intestine, cancer cells, I think, and obviously fibroblast cells. So, um, it's interesting. I think it's interesting to see if, since the sort of organization of the, I think my AirPods died. Okay, hopefully you guys can still hear me. My AirPods died, so we'll see how the audio turns out. Okay, so if the whole cell is polarized, why can't the nucleus also be polarized? Is basically what I'm getting at. So, um, to and we see this reorientation, right? So like the cell will make sure that to get through small and tight spaces like other cells or filters, right? It has to put the smallest width of the nucleus to go vertical or go through that passageway. So something defines the apical and basal ends of the nucleus, I think. So this aim is here to see whether the nuclear envelope polarizes during migration, and then how this polarization aligns with cellular polarity. So, does it fit in with the rest of the polar elements in a cell? Is basically what I'm trying to say there. Um, Again, same exact setup, live cell imaging, confocal microscope, um, fluorescence, actin, sun 1 proteins, and then the perinuclear diene, or the BICND something that you remember. um, so one cell, like one side of the nucleus, I'm thinking has a different non-random distribution of these sun proteins. So either the sides of the nucleus, like the small edge or small width, maybe there's sun proteins on those two sides or just one of them, or the like apical and basal ends have more sun proteins than others, and maybe just the apical one has it, or maybe just the basal one has it. So there's a bunch of different analyses or softwares that you can use to find like basically correlations between this specific spot that you marked in the nucleus and then the rest of the cell movement. So this is just an example. The mean squared displacement thing is just something I saw. And so let's say there's no pattern, or like the sun proteins are randomly distributed around the nucleus. This would suggest that nuclear rotation and directional positioning are not determined by the location of the sun proteins or of the LINC complex in general. Or it would support the hypothesis that the nucleus is not polar, right? Okay, so the impact here, I said earlier, it can like inform therapeutic interventions, and if we can like mess with cellular migration, that has a lot of different therapeutic uh, potential. whether it be wound healing or metastasis or other polar cells that move. You think of neurons or, yeah. So we should do this. You should fund me, basically. And so, yeah, that's it. Thank you for listening. If you have any questions, please put them in the comments. Um, I This project was put together in about 2 weeks, and so it's not super in depth. It's not like I don't know too much about it, is basically what I'm saying. But I'd like to learn more because of this section in the program, this cell and development biology section. I'm pretty sure I'm going to join that department at the end of the year, but we'll see if things change. In the spring semester, I'm going to rotate with my facilitator for the section and do some of this imaging myself. So I'm really excited. We'll see how that goes. Thanks.